So the control of skeletal muscle activity is at the neuromuscular junction. There is a special intercellular connection between the nervous system and the skeletal muscle fiber, and it controls calcium ions, one of the body electrolytes, known here as a Ca with a plus, so it's a positively charged ion, to release into the sarcoplasm. Alright, so here we have the diagram taken from your books, and we can see here that we've got what is called a motor neuron or a motor nerve in the top part of the red band of the picture, and it moves down, moving down its action potential until it meets those blue ends, which are the motor end plate, which is attached to the outside of the muscle cell. When it attaches to the outside of the muscle cell, you can see that the little red baubles are released and they're taken up by the actual cell membrane. We're going to where it says see below, we're going to the next picture, the next slide. So here we have the cytoplasm from the synaptic terminal contains vesicles with molecules of acetylcholine. And acetylcholine is a very common neurotransmitter, a chemical that is released by the neuron, the nerve cell, to change the permeability of another property of another cell's plasma membrane. And the synaptic cleft and the motor plate end contain molecules of the enzyme acetylcholine esterase, which helps to break down acetylcholine. So again, the muscle will be able to contract again. So what we're looking at here is we're looking at the little blue circles. So they are vesicles, and within the vesicles is the red chemical acetylcholine. Then we've got the line of blue, which is the end of the nerve cell. And then we've got that sort of fawny coloured part with the little moon shapes and the little purple square bits. So that is the sarcolemma, the actual surface of the muscle cell. So that's a microscopic view of the end of the nerve cell and where it touches the actual muscle cell. So the vesicles are released in the next picture. When the action potential comes through, when the action potential passes, the vesicles are released and the acetylcholine is released, and that causes a change in the motor end plate of the sarcolemma, causing a change in the permeability to sodium. And then the acetylcholine meets its receptor site, so sodium moves into the cell. Extra acetylcholine that's not used is quickly broken down by acetylcholine esterase, which are those little moons, so that the action potential can then occur again.